Hi, my name is Rachel McCarley. I am a senior honor student at William and Mary, and this is my presentation for my thesis to the Spring 2025 Honors Research Symposium. So my project is titled HerbertWM and Beyond, a dual approach to understanding and engineering phage host range. So a little bit of background, first of all. The global problem of bacterial antibiotic resistance is very well known, but bacteriophage are not as well known. Bacteriophage are viruses that can infect bacteria. They're not able to infect humans, animals, whatever, specifically bacteria. So they provide this really interesting potential way to address this issue of antibiotic resistance by completely circumventing it with a completely new approach. The issue is with something like phage therapy when you're using applications in medicine in the lab, but especially in medicine with patients, is this need for precision and control to know which bacteria species and strains these phage can and can't infect whenever you're putting these into a living being. So the big research question here is, can we predict, design, and engineer phage host range? And on the left here, for context, is a transmission electron microscope image of a novel mycobacteriophage that was discovered and isolated in the lab at William and Mary. Just pretty cool. So in terms of methodology, I use a dual approach. So the first half of it is a bioinformatic approach, which involved building a database of bacteria-phage interactions. So positive interactions where the phage can infect the bacteria, or negative where it can't. And these were all experimentally verified. And so alongside these interactions, I obtained the genomes for the corresponding bacteria and phage. And all of this was put through a machine learning model and training the model to predict whether or not any given pair of bacteria and phage input into it can or can't infect. So on the left here is kind of the pipeline for usage of the model. So you would take a bacteriophage raw genome sequence, AGTC nucleotides. Those are encoded into binary zeros and ones, and then they are passed through the machine learning model that has been trained on my data set. And then it spits out a number. So it's somewhere between 0 for no, 1 for yes. Basically, it gives you a probability of whether or not that phage that you have input is able to infect that unknown bacteria pair. So on the right, you can see here a plasmid, which is representative of my engineering approach. So my engineering approach uses a mycobacteria targeting system by Shankerman et al. And it involves unmarked gene deletions in mycobacteria. So using this system, I constructed a vector for gene editing in the phage HerbertWM. In terms of results, on the left here, you can see this little chart, which gives you a list of 6 phage that I tested, that I ran through the model. And they're in the same family as this phage, HerbertWM, which is why this is relevant. And so I put them into the machine learning model, got a prediction value, and then I actually tested these against Mycobacterium achiense, the native host of phage, HerbertWM, to see whether or not that prediction value by the model was correct. And it was. So not only does this validate the model with actual experimental data in the lab, it also contributes to the body of literature and the training data to make things more robust and generally increase the accuracy of approaches like these for future use. And in terms of results, 70% of the phage bacteria pairs selected by the machine learning model were able to infect or were positive for infection. The right here is something small and representative of the engineering approach, but here are bacteria colonies that contain this constructed PYS2 GP3 gene deletion vector that I constructed. So in terms of future directions, I will proceed to test this vector to characterize the phage HerbertWM, and then later alter the vector to swap in a new gene to try to expand the range of bacteria this phage can infect. Thank you. And I would like to thank the Charles Center again for providing funding for this project.